All right, let's talk about differential gene expression in development. So, development is just the process of a multicellular organism starting off as a single cell and then going through a series of changes in successive forms that will eventually characterize its life cycle. So, we have three different processes we'll talk about today. Determination, that's going to be what determines the fate of a cell. So determination is going to be the cells which may receive signals to determine its eventual cell type. It's what's gonna happen to it when it grows up. And then we have differentiation itself. So that's going to be the cell actually changing in its gene expression that would allow it to differentiate from other cell types. And then lastly, we have morphogenesis. So that's just going to be the reorganization and potential elimination of cells through apoptosis to help better form or create the morphology of that tissue, of whatever tissue we're talking about within that multicellular organism. All right, so let's start off with determination. So determination is what's going to determine the cell fate, and one way you can get determination is through cytoplasmic segregation. So that's when there's an unequal cytokinesis, giving cells polarity. And by unequal cytokinesis, what I mean is when the cell divides, you're not going to have an even distribution of all of the cell contents throughout the entire cell. Sometimes you'll have a gradient or a different concentration of certain molecules in one end of the cell versus the other. And so in this case, we have your original cell, so an unequal distribution of cytoplasmic component. We could even say these are like transcription factors, maybe. And so you get a cell division, and you get a kind of an equal division down the middle, where you actually split we'll say their transcription factors evenly between the two. So it's retained as the cell divides. But let's say that cell divides again, and now what we have here are these two upper cells which have none of that transcription factor, whereas the cells down below have different concentrations of transcription factors. Those transcription factors which may contribute to differential gene expression in these cells versus these cells. So we can have cytoplasmic segregation, which leads to determination. Now we have another type or another way that we can get that determination. So we have induction. So this is going to be something exogenous, something from the outside of the cell, and it's going to involve cellular signaling. So we could have a central cell. We'll just say that this is the inducer. It's going to be the inducer cell, or the cell that produces the inducer. So what it's going to do is, maybe we'll say that it releases some kind of signaling molecule, which would cause a cell to eventually differentiate into a different cell type. So one is the original cell produces an inducer, releases that inducer, and it's probably going to release an inducer in some kind of gradient. The cells that are closer to it are probably going to receive more of that signal, whereas the cells that are maybe a little bit further away might receive less of that signal. And that's going to have an effect on what eventually happens to the neighboring cells. So two, the diffusion of the inducer forms a concentration gradient. Again, that's higher concentration closer to the original cell, and then a lower concentration towards the outside, or further away from the cell you get, the lower the concentration. And then you have here this cell here, which has an even distribution of receptors, and it has a lot of ligand binding to it. So this cell receives many inducer molecules that bind to the many receptors. However, this cell that's maybe a little bit further away has an equal amount of receptors. However, because it receives less of that inducer, well, it receives very little inducer, and it may not differentiate in the same way as the other cell that was closer. So here we have the inducer binding results in a transcription factor activation or translocation into the nucleus. So we'll get some kind of signal transduction or signal cascade from that receptor, eventually leading to a protein, bumping into a protein, bumping into a protein, maybe the synthesis of a protein, or not the synthesis of a protein, but a morphological change of a protein, which eventually slips into the nucleus and causes differential gene expression. And that cascade is going to eventually lead into this cell becoming a different cell type than the cell that didn't receive as much signal. So again, we've got cytoplasmic segregation, which involves internal signal, and then we also have induction, which involves external signal or exogenous signal. Now we have differentiation. So once the cell has received its signal, now it can start expressing different genes. And so here, the next step is the inducer binding results in transcription factor activation it results in the in transcription factor activation or translocation into the nucleus. And so once you have maybe new transcription factors entering the nucleus, well, that's going to maybe promote some genes to become transcribed and therefore translated, whereas the other cell had not received those transcription factors might not express those same genes. So it's going to increase the affinity of certain genes to be expressed within this cell, those genes that may cause it to differentiate from other cells. So the transcription factor may bind to a promoter, which, remember, this cell did not receive as much signal, therefore you're probably not going to get the same kind of transcription factors activating, those genes. It's going to bind to a promoter and it's going to activate gene transcription, and eventually you get transcription, and then you eventually get translation, and now this cell is pumping out different proteins from this cell, which has not received enough signal to differentiate. So the protein encoded by the gene stimulates cell differentiation. So it could be a self-propagating event where this cell receives signal to transcribe and then translate proteins associated with an internal proteins associated with further differentiating the cell. So this protein could help stimulate cell differentiation, whereas this one, which didn't receive signal, it's not going to be receiving that transcription factor binding to its DNA on the promoters for those specific genes, and therefore it's not going to produce those proteins involved in cell differentiation. So some of the results of differentiation are going to be result in specialized biochemistry. So this might alter its biochemistry, its structure, as well as its function. So differentiation is just the act of turning into a new cell. Now, last we have morphogenesis. So that's going to be the cell response to differential gene expression to form patterns that result in spatial organization. So if you look here, you've got some kind of mammal, or you could just say multicellular organism that has digits. And so here you start off with like the baby stages of digits, and you have these interdigital tissue in between those digits. But over time, what might happen is you'll have cell signaling within this tissue, but you're going to have differential cell signaling depending on where it's located, but depending on where the cells are located. And so maybe around the digits themselves, well, they might be releasing transcription factors to neighboring cells around the digits in order to induce some kind of apoptosis. And then these cells within the interdigital tissue might be releasing transcription factors to further enhance maybe the defense or the non-apoptosis of the digits, and then but also to enhance the apoptosis of its neighboring interdigital tissue. So cells undergoing apoptosis and eventually will form digits in between, form digits through that interdigital tissue apoptosing, or apoptosis would be programmed cell death. Okay. So let's talk about some organ identity genes. So this is an example of a flower, and we're talking about 3 different types of genes. So 3 different types of genes are going to be involved here. Here's the original flower, and we'll just say gene type A, gene type C, and gene type B are all important factors. The thing about flowers is they'll have, you could say, 4 different layers. They're organized into 4 different whorls. So whorl 1, whorl 2, whorl 3, whorl 4. The very outside whorl is the sepal, so that's the green kind of sheath thing. And then whorl 2 is going to be where your petals will emerge from. And then whorl 3, that's where you're going to have your stamens emerge from. So that's going to be you could say the male mating type, the organs that will eventually produce pollen, for example. And then lastly, whorl 4, which will be central, that's going to be your carpel, or you could say the female mating type organ. Now, in order to express your sepals, all you need is gene A. Now, in order to express petals, you need both gene A-type genes, as well as B-type genes. So in order to meet that criteria for petal formation, you need both of these present simultaneously. Now, in order for your carpels to develop, so that's on whorl 4, you need your C-type genes. So C-type genes, that's the only criteria. As long as you have your C-type genes, you're going to develop carpels. Now, in order to develop your stamen, or the male organ, you're going to need both C and B type genes. So now let's say you introduce a mutation to this flower and you remove all B type genes. What you're essentially going to do, because you're still producing A and you're still producing C, is you're only going to eliminate the petals and you're going to eliminate the stamens. So as a result of eliminating the B type genes, so mutation of class B, you'll get no petals, you'll get no stamens, and instead you actually get those whorls replaced by carpels and sepals. So what you'll have is, in whorl 1, you'll have sepals, in whorl 2, you'll get sepals, and then in whorl 3, you'll get carpels, and then whorl 4, you'll get carpels. So they're actually replaced. Now let's say, I don't know, you remove your type A genes. We'll remove type A, and so all you have are B and C. So you're going to meet the criteria for your carpel development. You're going to meet the criteria for your stamen development, but you're not going to meet the criteria because, remember, A is necessary for your sepal and your petal development. So as a result, what happens is, let's say you replace the A with C, and what you're going to get is on the very outside, you're going to actually make carpels, and then just inside of that, you're going to create stamen, and then you'll have stamens again, and then you'll eventually have carpels. So you can kind of see how this could be used as kind of like the central map. Let's just talk about another one. Let's say you remove C, the C-type genes. Well, what's going to happen is you're going to have all A and B. So you're going to meet the criteria for your sepals, and you're going to meet the criteria for your petals. And so what happens is you'll eventually create two layers. You'll have sepals, and you'll have two layers of petals, and then you'll have sepals again. So those are organ identity genes. Now let's talk about your Hox genes or your homeotic genes. And so what these are going to do is, kind of like we talked about earlier, these genes are going to spread in a gradient, inducing cells to undergo differential developmental programs. So here we have source cells, so these green cells, they're going to release, you could say, the homeotic genes in a gradient. So you're going to have a higher concentration around this blue area and a lower concentration around this white area, and zero concentration around this red area. So here, this is a chart showing the different thresholds you must meet in order to induce cell differentiation. So here you have on the y-axis the concentration of the morphogen, and then you could say on the x-axis you just have the spatial arrangement of cells, so the different layers of cells. So around 1, you're going to have a very high concentration of that morphogen being released, or the homeotic genes being released. And what that's going to do is it's going to cause a differentiation in that area. So the threshold for inducing the blue fate, so these cells will become blue, that are close by those source cells, the ones that are actually releasing that morphogen. Now, if you have a lower concentration, that's going to be the threshold for inducing the white fate. So you got the blue, which received a lot of cell signal, a high concentration of that cell signal, and then you have your white cells, or the cells that receive a moderate to low amount of cell signal, which will not differentiate into blue cells, but instead differentiate into white cells. And then you have the red cells, or the red fated cells, which receive no signal, we'll say. So the position of each cell is defined by the concentration of the morphogen. Now the thing is, when these blue cells receive that morphogen, for example, and as they start to differentiate, they might be releasing cell signal to their neighboring cells. So let's say we have a moderate amount of that initial signal being received by these potentially, or these destined to be white cells, and then you have these blue cells that are releasing a gradient of morphogen as well, or transcription factors. Well, that might enhance the differentiation of these cells. Maybe these cells require, two different cell signal, and what that's going to do, or having those two different concentrations of transcription factors may do, is allow for cells to, in a sense, identify where they're located relative to other cells. Now, these white cells might start to become white cells, and as a result, start releasing transcription factor to these neighboring red cells and these neighboring blue cells, maybe reinforcing their cell fate. And so, as a result of that gradient, your cells are kind of able to identify where they sit in the larger morphological sense relative to other cells. So let's talk about something a little bit more tangible. So this figure on the right uses fruit flies or Drosophila as an example, and they also have Hox genes, and they're actually arranged on their chromosomes in a linear sequence that kind of reflect the order of expression in the actual embryo. So here we have, your head region, what would eventually develop into thorax region, and then an area that would eventually develop into the abdomen region. Now you're not going to necessarily, or hopefully, you're not going to get eyes growing in the thorax or wings growing on the head. And that's because they actually form their morphology in the way that we described earlier, where we were talking about the morphogen gradient from releasing transcription factors. So I'm actually going to go back to the abstract again, and I'm going to use an example here. So we'll say our origin cells are red. Here's our origin cells, the original cells. And during embryo development, let's say they release a certain cocktail of transcription factors to the neighboring cells. So these two columns here will be neighboring, these two columns here will be neighboring. And what that's going to do is that's going to cause them to differentiate into these yellow cells. Now these yellow cells, when they receive that transcription factor, that might signal them to release their own type of transcription factor. Not the ones that the red cells release, but the ones that the yellow ones will now release. And so let's say they release transcription factor in both directions. Now they're going to actually signal to the cells that originally sent them the first signal. And they're also going to send signal, within proximity to these neighboring cells on this side over here. What that's going to do, and we'll just say that these release their own transcription factor, that's going to cause these cells potentially to turn blue. We just say they'll turn blue, for example. However, these cells that have already once received signal, or not once received signal, the ones that originally sent the signal, which are different cells from the yellow cells, are receiving signal from the yellow cells. their own transcription factor from the yellow cells, which is going to cause them to become orange. Now, these blue cells, when they have received that signal from the yellow cells, they're going to release their own transcription factor, interacting with their neighboring cells. So we'll just say that there's some hypothetical neighboring cells, on the outskirts here. But what's going to happen is these yellow cells that originally sent signal out to cause these blue cells to differentiate are now going to be receiving signals from those blue cells, causing them to turn green. So that's back on abstract, but you can kind of see how, or hopefully that would paint a better picture of how your cells within, or you could say your tissues would develop in relation to the proximity of other tissues within an organism. Now, fun fact, the same Hox genes that determine the Drosophila embryo development are actually the same type of Hox genes that will direct the pattern of our own nervous systems.